Bismillah ar-Rahman Rahim. We start the November 2022 papers, which are the latest papers that we have, and this will be the paper 1-3 MCQ for the new syllabus 2023 syllabus, 2022 syllabus, sorry. As we all know that the timings have changed. Now this will be 1 hour 15 minutes. Usually it used to be for 1 hour, and this is the variant 1-3. And you know the rule, that's 40 questions, answer all questions. For each question there are 4 possible answers, and you write with a soft pencil and write your name, center name. Do not use correction fluid, do not write on any barcodes, and you may use a calculator. Now the rule about doing MCQs is that you read the question twice, and you try to see what is the main focus of the question. The electron micrograph shows a chloroplast from a tobacco leaf. If the actual length of this chloroplast measured along XY is 10 micrometer, what is the magnification of the image? So you measure it at 6.3 centimeter or 63 millimeters. So you convert it into micrometer because it is equal to 10. So divided by 10, what is the magnification? It is 6300. So the answer is D. But basically, how do you do it? You convert it into the same, you measure this from X to Y, you measure it in millimeters, and then you convert it into micrometers, and then of course you divide it by the original. So magnification is equal to image over original. Now question 2, some cell structures are listed: endoplasmic reticulum, Golgi body, mitochondria, and chloroplast. Which cell structures do not contain chloroplasts? Do not contain cristae. You know the cristae is what? The cristae is the folds created by the inner membrane in the mitochondrion. So matrix inside the mitochondria created by the inner membrane. So anything which doesn't have 3 in it, so it says do not contain cristae. Mitochondria contains cristae, so it has to be 1, 2 and 4, and that is B is the answer, because it can't be 3. So this was wrong, and then you have to narrow down to B and C. 1 and 2 is Golgi and endoplasmic reticulum, and those contain cisternae. Now I want you to be very clear about what is cisternae and what is cristae. They're totally two different things. Now as you can see here in the Golgi apparatus, we have transport vesicles, and then we have the lumen, and then we have the newly formed vesicles, and then we have the secretory vesicles. And in this you can see these infolding, these is called the cisternae. So you must know the difference between cristae and cisternae. Now you can look at this table. This is cristae. So cristae is where each of the partial partitions in a mitochondrion formed by the infolding of the inner membrane give a signature wrinkled shape to the inner membrane of the mitochondrion, contains proteins including ATP synthase and a range of cytochromes. While on the other hand, cisternae is the flattened membrane disc of the endoplasmic reticulum and Golgi apparatus. Endoplasmic reticulum, cisternae. Golgi apparatus contains around 33 to 20 cisternae, then contain different types of enzymes inside them. Now coming to question number 3, what you have to understand is which letter represents cell structures where mRNA may be found. So in the nucleus, yes, DNA to mRNA, so this will be transcription. DNA to mRNA, so this will be transcription. So yes, in the nucleus also. In the cytoplasm, why? Because in the cytoplasm you have the free ribosomes. And the free ribosomes is where the mRNA is going to get attached to the free ribosomes in the cytoplasm. So free ribosomes. And then of course, if you look at mitochondrion, why would there be mRNA there? Because in the mitochondrion, if you remember, there is a loop of DNA and they are free, and there are 70 S ribosomes, so they would also be there. So here also, and here also. So that is why the answer is B. So the answer is B to this is for this reason which I have just explained it to you. Then coming on to question number 4, which is the correct for typical prokaryotic and prokaryotic cells and mitochondrion. Now, when you look at this question, which is row is correct for typical prokaryotic cells and mitochondrion? Well, you know prokaryotic cells and mitochondrion have nearly the same size. So this is wrong. Then 70S ribosome is present in prokaryotes and bacteria, so this is wrong. Circular DNA is present in both, so this is wrong. And the only answer is C, which is correct, is peptidoglycan. Prokaryotic cells wall is peptidoglycan, and mitochondria, it is not. There is no actually cell wall in mitochondria. So another difficult question, I think, if you are not very clear on it, I just want you to see a diagram. Now this is a very good scale diagram about the different sizes that we have. So if you look at bacteria and mitochondrion, they're both the same size. Then the animal cell and plant cell are the same size. Then, sort of you look at all the organelles with another one diagram which you need to see about the sizes of the different organelles. Now let's look at question number 5. Which statement is correct? A virus is composed of a protein coat that surrounds RNA or DNA. Yeah, that's correct. But then why is B wrong? This is what I want to talk about. Suberin and cellulose are found in all plant cell walls. No, suberin is found, if you remember, in the Casparian strip. Cellulose is found in plant cell walls, not suberin. Suberin is found in impregnated in the only in some cells, which is in the Casparian strip, which is the endodermis. Then plasmodesmata and centrioles are found. Centrioles are not found in plant cells. Only plasmodesmata are found in plant cells. Then prokaryotic cells contain an endoplasmic reticulum to synthesize proteins. Now, please remember prokaryotic cells do not have any membrane-bound organelles, so they would not have any endoplasmic reticulum. Now coming to question number 6. The Benedict's test and ethanol emulsion test was carried out on a sample of biological molecules. The solution became brick red during the Benedict's test and cloudy during the ethanol emulsion test. So Benedict's test brick red means reducing sugar present, and ethanol emulsion test, if it becomes cloudy, means fat present. So which molecules did the samples contain reducing sugar and lipids? Okay, so this is the answer A. B is wrong. Why? For proteins, you do the biuret test. For Benedict's, you do it only on reducing sugar. Starch, you do the iodine test. Protein biuret test, Benedict's test. And non-reducing sugar, you do sucrose, and then of course you do HCl hydrolyze and all that. And lipids is the ethanol emulsion test. So that is why the answer was A and the other are wrong. Now coming to question number 7, which diagram is correct for alpha glucose? Now the answer is A, but then why is B, C, and D wrong? That I am just going to explain to you. Now if you've looked at my other videos, I've always said, easy way to remember alpha glucose is alpha below, beta above. Now what is below and what is above? Alpha, the OH is below on carbon 1. This is carbon 1, this is 2, this is 3, this is 4, this is 5, and then this is 6. So if you know the carbon numbering, then you know on the carbon 1, the alpha glucose, the OH is below. So OH is below in only these two. This is wrong because the OH is above and the OH is above on this. So these two are wrong. Now here, what do we have it wrong is we've got this wrong here. So alpha below, beta above, ABA is the mnemonic to remember beta glucose and alpha glucose, which is something which is part of the syllabus, which you need to understand and remember. Now coming to question number 8, which reaction correctly represents one way sucrose can form? Now alpha glucose plus fructose. So and it makes sucrose plus water. So this minus water and this minus water is wrong. Now, so you're left with A and C. So in C there's alpha glucose and beta glucose. No, I'm sorry. One is glucose and one is fructose in sucrose. So the answer is A here. Now I've shown you the diagram of it as well. So alpha glucose and then how the glycosidic bond is going to be formed and how a water molecule is going to exit. And that is why it has to be plus water. A water molecule has to exit. This is a condensation reaction, not a hydrolysis reaction. The diagram represents part of an alginic acid molecule. And alginic acid is a polymer found in the cell walls of brown seaweeds. Which statement about alginic acid is correct? So you just have to look at this diagram. Nothing else. You don't have to know any prior information. Alginic acid is a linear polysaccharide containing 1,6 glycosidic bond. No, I can't see any 1,6 glycosidic bonds because then it would be between this and this. So 1,6 is 1,4. Alginic acid is formed from two monomers joined by 1,4 glycosidic bond. Yes, two monomers. So this is one monomer and this is the other monomer joined by 1,4. So answer is B. But why is C wrong? Alternate monosaccharides are arranged in opposite direction. There is also seen in glycogen molecules. No, I mean we don't see any opposite directions here. Plus hexose sugars and water molecules are joined by condensation to make. No, we can't see that either here. So this answer was B. Then we come on to the 10th question. Which features are correct for triglycerides and phospholipids? They have ester bonds between glycerol and fatty acids. Fatty acid chains may have single or double bonds between carbon atoms in the chains. The fatty acid chains are non-polar and hydrophobic. Saturated fatty acid chains allow closer packing of the molecules than unsaturated fatty acid chains. Now, when you look at it, all these are very correct. So, if all of them are correct, then the answer is A because 1, 2, 3, and 4 they do have ester bonds. They have single or double, they can be saturated or unsaturated. Whereas triglycerides and phospholipid can have either saturated or unsaturated. The fatty acid chains are non yes in both of them, this case. And then saturated allow closer packing of the molecules than unsaturated ones. So the answer is A. Question number 11. A scientist studying the structure of a protein reported that it consists of two polypeptide chains joined by disulfide bonds. Which feature of protein structure does this describe? Well, if it's two, we always said if there are two or more polypeptide chains, then it is a quaternary structure. So, if in the question they're telling you there are two polypeptide chains, then that means it's telling you about the last configuration of the structure. First, the primary structure is the number and sequence of amino acids. Secondary structure is the alpha helix and beta petal sheet. Tertiary structure then contains further four bonds. So now this was to confuse you when they said the disulfide bonds. So the disulfide bonds are disulfide, hydrogen ionic, and the van der Waals forces of the hydrophobic hydrophilic interactions. So when they said two polypeptide, then it's two or more, that means it's the quaternary structure. Question 10. Some molecules fluoresce when illuminated with ultraviolet light. Changes in the structure or shape of such molecules can change the intensity of the fluorescence. The enzyme tryptophan synthase fluoresces bluish green because of a phosphate group in its active site. So there is a phosphate group in its active site, PO3. There is a phosphate group in its active site. The enzyme catalyzes the reaction. Serine plus indole gives you tryptophan. So serine fits in here and some indole fits in here and you get tryptophan. So adding serine to the enzyme increases the intensity. So if you added serine to the enzyme, it increases the intensity of the fluorescence. But when indole is also added, when indole is also added, this indole is also added, the bluish green decreases in intensity. Okay, so this causes a decrease in intensity, and serine increases the intensity. So this increases the intensity and that decreases the intensity. So serine and indole result in. So we have a problem here. So okay, we have indole here and serine here. So this is serine and this is indole. Let's talk about that. What may be concluded from these observations? Serine and indole attach to the active site of the enzyme. Yes. An enzyme serine complex is formed. Yes. An enzyme serine indole complex is formed. So that's why the answer is A. 1, 2, 3. Question 13. Yeast contains the enzyme catalase, which catalyzes the breakdown of hydrogen peroxide to water and oxygen. So yeast was added to a solution of hydrogen peroxide and total volume of oxygen released was recorded every 30 seconds for 2 minutes. All other variables were standardized. The data is shown in the table. So in 30 seconds, we had 157 cm cube of oxygen, then 251, then 283, and then 285. So in the beginning, we had 157 cm cube, then it went up to 251. So at least another 100 has been added, and then only 56 and 34 have been added, and then 283 to 285 has only been 2. So something's gone wrong here. As time has gone by, less oxygen is being produced. So in the initial stages, oxygen was produced was more. So what explains the pattern of data? Now the answer is of course D, but the volume of oxygen released decreases as more substrate is converted into product. Now that's very obvious, 283 to 285. Now why are A, B, and C and D, A, B, and C wrong? A, the rate of reaction increases as more enzyme substrate complex of no, the rate here has decreased. The rate of reaction increases as the enzyme reaches the maximum velocity. No, we haven't got anything. We haven't got it at a constant rate as yet. Then the volume of oxygen released decreases as they no, it's never decreased. It's not decreased. The rate has changed, but it has never decreased. So that is why the answer was D. Then coming on to question number 14. The Michaelis-Menten constant Km is a measure of the affinity of an enzyme for its substrate. The higher the affinity, the lower the Km. The lower the affinity, the slower the reaction will be. At Km, half the active sites of the enzymes are occupied by the substrate. Now, which statements about Km are correct? Now, either you know Km or you don't know Km. So don't make any guesses. Either you know this or you don't know this. So if you look at the answer now, it's the A is the answer because all of these are correct. The higher the affinity, lower the Km. Yes, that's correct. The lower the affinity, slower the reaction will be, and actually the Km is going to change. At Km, you have to remember what is Km is half V max. The value of the concentration of the substrate at half Vmax. So at Km, half the active sites of the enzymes are occupied by the substrate. Yes, that's correct. Okay, now coming on to question number 15. Which features are correct for active transport and facilitated diffusion? The movement of molecules in ion depends on ATP. No, facilitated diffusion does not depend on ATP. It's only active transport which depends on ATP. Then there are specific for one type of molecule or ion. They use membrane proteins. Now, what do you think is the answer? Now, naturally, the answer is C. Why is it C? Because one, the movement of molecules in ion depends which features are correct for active and facilitated diffusion. So this is not, so this is wrong because only ATP is required for active transport, not for facilitated diffusion. They are specific, yes, always this is channel protein specific for only one type of molecule or ion. And yes, channel proteins are used for active transport, which has an ATP binding site, but the one for facilitated diffusion does not, is also a channel protein involved, but it does not have an ATP binding site. Now, coming to question number 16, the diagram represents two solutions, P and Q, that were separated by partially permeable dialysis tubing, partially permeable dialysis tubing, solution P and Q. Now, molecule 1 is going to be in this direction. Why? Because there are more of molecule 1s on this side. You can count them and see them. There are more. So molecule 1 would move in this direction, right? Now, let's look at another one. Now, let's look at molecule 2. Now, if I'm going to give it another color, molecule 2 has the same number on both sides. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8. So there's going to be no net movement of molecule 2. Now that is only here. So that is how I would have said the answer was B to this. Because, but then of course in D also this is the answer. So how would I say it's not D? Because there's one incorrect one. You see molecule 3 Molecule 3 was P to Q. Why was molecule 3 P to Q? Now let's look at that. I'm going to give it another color. Molecule 3 would, let's give it this color. Molecule 3 is going to be from P to Q. Now let's look at it. This is 1, 2, 3, 4, 5, 6. And there's 0 on this side. So it would naturally move from this side to this side. So that is why molecule 3 would net movement from P to Q. And net movement from Q to this thing would be molecule 1. So this is how you had to do this question. It was a pretty confusing question. Now coming to question number 17, a student weighed a cylinder of potato, then put it in a test tube containing a salt solution. So okay, we took a test tube and it contained a salt solution, and you put a piece of potato in it and you put it in this salt solution. Always do a little diagram to explain things to yourself what we mean. Then the potato cylinder was removed from the salt solution after 1 hour and it was blotted dry and then reweighed. The student recorded the potato had lost mass. So that means water has gone out of it. Water can only go out of it. It's a question on osmosis. Which row shows the correct explanation for the results of the student? So water potential of the potato cell before soaking compared to the after soaking, it had to be higher. So it had to be either A or B. So it either had to be A or B. Now, condition of the potato cells after soaking would then naturally be plasmolysed. So if they are plasmolysed, then answer would be A. So the fact that you put the potato in and lost the mass, then of course it means water is moved out of it. Now coming to question number 18, which row shows the correct number of each component in a single chromosome at the start of telophase of mitosis? Now this is when it was joined up, and here in mitosis now they will be separated. So in each question, what do we have? We have only one centromere. Polynucleotide strands will be of course two, like A, T, G, C. So there will be two. And telomeres, there will be two. Naturally, there will be two telomeres. This is one telomere here and the one telomere here. So that is why the answer was A. What you have to understand is that polynucleotide strand are two. Centromere is only one. So this is the only centromere that we have here for each. This one has one and this one has one. And telomeres, of course, as I've told you, would be two. Question 19. The photomicrowave shows cells in various stages of the cell cycle. In which stage does semi-conservative replication of DNA take place? Why C? This is interphase. Now, if you remember, in interphase is G1, S, and G2. In S phase of interphase, does the DNA replication take place semi-conservative? So what is this? This is metaphase. Just a quick revision. This is anaphase and this is prophase, when they've condensed and they've become thick and they're still in the nuclear membrane, is still starting to disintegrate or has going to is disintegrated. We can't really see much of it. Now coming to this very long-winded question, I always, when I read such long questions, it gives me a very tough feeling. The diagram shows the epidermis of human skin. This is formed by 4 layers of cells. Okay, fine. Well, we're going to have to read all this. Cells in each layer have specific functions which are outlined on the diagram. As the cells are produced, they move upwards as shown by the arrow. So they're moving upwards like this. So let's start from the bottom. Basal cells divide by mitosis and differentiate into the keratinocyte cells, which move up into the layer above as they are produced. So which row shows cells that are acting as stem cells in the skin epidermis and an organelle needed by the keratinocytes? Now, the cells that are acting as the stem cells can only be the basal cells. I would have read this first part and I would have figured this out, that this was only going to the basal cells. So other two options are wrong. Basal cells and keratinocytes, at least I feel they're wrong. So the basal cells divided, they move up, so they're moving up here. Now this is called the keratinocyte cell. Begin the synthesis of the protein keratin and glycolipid. Protein, of course, would require ribosomes, and glycolipids would require the Golgi body to prevent water loss from the skin. Then the keratinocyte cell continue to synthesize keratin and glycolipids to waterproof the skin. Cells in this layer are dead and eventually break away from the skin. Now, which row shows cells that are acting as stem cells? One, organelles needed by the keratinocyte. Now, of course, the answer is B. Now, why is the answer B? Because it's basal cells and it's the Golgi body, glycolipid. Glycoprotein, glycolipid is the Golgi body's work. Centrioles have no work actually. We don't even know what they was required in cell division, and they are helping in the spindle formation. So that completes this question number 20, and I will continue this in the next video, the rest of the questions of this, and I hope this is helpful. And please do leave a comment, and please do let me know if I can improve anything further so that this becomes a better exercise for all of you so that you can take your exams in the coming months.